I'm very grateful to be here. It was many years ago I came here as a scholarship receiver because I had no money. And at that time, I have no money again, by the way, for this. But at that time, I came up with the ideas that I'll talk about today. So I thought we had a great talk, can you switch it over, about the dangers of omniscience just now. And so I'm going to follow that, it's kind of a debate, with the bliss of ignorance. So he's the omniscience, I'm the advocate for ignorance. And I was just really shocked as a forensic scientist to hear one of my favorite aphorisms about the absence of evidence, that the absence of evidence is not the evidence of absence. And so I'd like to twist that around. I study absence, I also study evidence. And so this talk is about kind of the blessings of absence. And it's the story of you, because we all started out the same way. We were absent, a potential, a missing combination, a group of components not yet combined. Then in a moment that is aptly called conception, the parts of you came together, and there was no going back. In terms of information, once we occupy a space in that Planck-length grid that is the whole universe of combinations, you can't be erased. Information is permanent. For living beings like ourselves, there is no reproduction. There is only recombination. And even if that happens at the molecular single base level, if we have a single base mutation, we like to think of it as de novo. It's not de novo, it's the movement of an electron that was already there, or it's the movement of a small chemical group. So let's skip ahead a few years from your conception to your thesis. You took a known body of work. They helped you build a scaffold out of it. These are parts that were assembled maybe 1,000 years ago, 100 years ago, 10 years ago, last week. And then they asked you to come up with a question. What is a question? Well, in order for your question to be a valid question, it has to be absent. You generated, probably through a fairly random process, a bunch of possible thesis questions. You went to the literature, and you had to find one that was absent. Once a change in information occurs and you publish your thesis, it is irreversible, and then it can be copied. So here's a brief history of the numbers that we use. And a curious thing is that these had to be copied by scribes or early mathematicians or business people. And you can see that once changes or transversions were introduced, they were copied into newer systems and they kept being passed on. So I wanted to look at this in a little more detail, again, when I had no money. And what I did was get a student, whose name is at the top here, Renea, sorry, and asked her to make a photocopy of an old book by Olds and Primrose, and then take that photocopy and make another, and another, and another. And this poor person did this. And what we saw is that you got all the types of mutations that you might expect in other processes, information processes, including the introduction of what appear to be new letters in the spaces. And then the translation of English to what looks like Cyrillic. So in biology, as I said, there is no reproduction, there is only recombination. My lab studies the transition from absence to presence and puts it to work in forensics and in medicine in developing drugs. And I think these studies can be extended to the areas of creativity and artificial intelligence. If we understand the basic rules of recombination in information systems, then we can have a chance to study and model mathematically creativity. I tell my students if I was starting now, I would study thought. Because although we're in the information age, you have no idea what a thought is made of. Does it have mass? What's its relation to temperature? Is it electrical, chemical, both? I believe that before we understand the substance of thought, we can look at the recombination that thought produces. And I'm inspired by a couple of authors, Claude Shannon, who looked at the recombination of letters and words, creating artificial English sentences, and before him, Gregor Mendel, the monk who humbly presented his work at the local garden club that described how the new theory of evolution worked. In 2007, Tim Anderson and I gave a talk here describing our search for Nullomers. These are the smallest string of missing DNA or protein sequences from all reported organisms. The term Nullomer is a play on polymer, the string of similar units that make up proteins and genes. And in 2007, we found that at length 5 amino acids, right, there are 20 amino acids, 3,200,000 possible combinations, there was no protein, there was no putative protein even, that had the sequences of 198 of those 5 amino acid combinations. We called those nullopeptides, being a short string of amino acids. And the odds of finding such short-length absence in such giant data sets was quite low from the models that we'd built from random distributions. But the question was it significant? Well, we're told that life began in an ocean 3.5 billion years or so ago. It evolves by chance. It's produced billions of species by simple recombination of 4 bases, A, G, C, T. And these are pictures from my lab, Bill Borland in my lab, who studies single-celled organisms called ciliates. And they're single cells, and what we were taught is, they reproduce and they're fairly clonal. But now we know that, no, they exchange DNA with other ciliates, and they can acquire genes from viruses. Here's one reproducing. So if life started out there in the ocean through some sort of recombination, this is Milo Leahy down in the south coast here, then why, in this 3.5 billion year experiment of combinatorial space, are there missing sequences? So I had no funding at the time that I came to my first meeting here in the 90s, but I wanted to jump into the genome revolution. I was teaching at a small college, and I was given a wonderful scholarship by this organization to come out. I was not so foolish as to think I could race against the luminaries who were studying all of the important stuff. And my father, who was a physicist, taught me, because he was also an inveterate junk collector, that you can find great things in trash. So I started looking at junk DNA. And as you know, most of the genome was called junk DNA at some point. It's only about 1% coding exons. 5% of them are endogenous viruses that have hopped into a sperm and egg and become immortally linked with us. And so I thought about studying that. But now we know that many important things come from this junk. For example, the placenta, the proteins required for that, came in on a virus and now have been incorporated. I needed something even junkier, something unnoticed, the dark genome, the outline of the tree of life where presence met absence. And I discovered that absence gives us something that presence cannot give us, the opportunity to create novelty. So what are the attributes of a creation as opposed to a simple novelty? Well, the patent office says it doesn't have to be de novo. You can take established patents, recombine them for a novel purpose, and you get a patent. And I think that's a great description of what a creation is, something that's recombined for novel use. In order to study creativity, we need to have a unit. I propose the AHA, the smallest unit of creativity. The AHA is certainly related to the bit. It's a unit of information. It conveys some measure of surprise. It can be a punchline to a joke. It can be the idea that you're talking about at the conference. Like information, the AHA has to be transmitted and received. If a symphony plays in a forest and no one hears it, there's no AHA. I've asked people over the last decade or so in many fields, what is the smallest unit of creativity in your field? And an actress said, breath. as a playwright, I appreciated that, and it shows me why acting is the superior art. I can write the words, they can twist the meaning. A breath can be a sigh. A breath can be a cough. A breath can be held. All of this can change the meaning completely. So what is the smallest unit of creativity in what you do? What is not derivative that you do? Well, we're all here because we don't want to be derivative. How would you measure that creativity? With the Nullomer approach, we first determine the units that can recombine, bases or amino acids, and then the smallest length at which absence occurs. There's no need to go beyond that if you're just looking for novelty, and it presents a manageable data set. Moreover, this is how our body detects novelty, unwanted novelty. The adaptive immune system of all mammals is a Nullomer-based system. Before birth, the genes responsible for all your antibodies recombined randomly like cassettes of DNA. I won't go through this, but these are just the genes and then the protein with the variable regions. The result is that each antibody-making cell will recognize and attack one short sequence of amino acids, curiously about the length of our Nullomer peptides, 5 amino acids or so. Since the DNA rearrangements are really random, there's a large number of self-attacking antibodies that are also made. In order to prevent autoimmunity, the body removes antibody cells from the fetus that identify a target in the fetus. Any antibody that binds such a protein commits suicide. What's left is the series of Nullomer hunters that you have to protect you against any novelty that enters your system. I think the same approach can be used to immunize data, and there are approaches that are doing that. Creation and destruction, that's where novelty is. In our bodies, it requires randomization. This randomization is fantastically restricted throughout your body's 30 trillion cells, except 2 places. The immune system, which destroys any novelty that comes in, doesn't ask any questions, just destroys it. And then your gametes, your gonads, that recombine information from your parents to produce offspring. So here's meiosis for you. It only happens in your gametes, where you swap pieces of DNA from your mother and your father and recombine, and that's that daughter nuclei line there. And then you cut the deck in half to randomize it further. And this is how the body produces both creativity and protection. Your unique combination of genes is the result of an ancient multi-step randomization that throughout evolution has produced better and better dice. As sexual beings, we especially do not reproduce, we recombine. And it happens, this recombination, without respect to fitness. In other words, evolution does not have a direction. The artist in our nucleus is scrupulously isolated from any selection bias. The dice are fair. They're locked in a double membrane away from the proteins that they code, unaffected by the novelty that we release as offspring. It is an investment strategy akin to long-term mutual funds that track the broad market, a blind trust in the growing economy of combinatorial space. And it requires symbolic units. DNA doesn't do anything. It is just a symbol, like letters, like numbers. It codes later for proteins. This symbolic, non-selecting mechanism of randomization is, I believe, what's necessary for creativity and our best defense against unknown threats. By studying what's absent, we've identified the shortest components of artificial life, sequences that are absent in all species called primes. You can think of them as prime real estate or prime numbers. For example, in the English language, there's only 2 letters that are also common words, A and I. This leaves you with a vast real estate of 24 single-letter prime words that should be trademarked immediately. I suggest for the sake of efficiency, we convert the most common words in each language to the single letter that starts that word. Think of the savings on ink, paper, electricity, and repetitive joint injuries. All right, so Nullomers sound like a party trick, something pretty fun, but are they useful? Well, the first Nullomer patent was issued to us to tag forensic DNA. Let's say you're arrested, you have to give a swab to the police. You ask them, are you going to be careful with this? They say, absolutely. They take it into the lab, and I will tell you that the technicians in the lab will process your DNA alongside with DNA from evidence in the crime you're accused of committing. You know we have polymerase chain reaction. It makes 1 billion copies of these things, and even a single molecule of your DNA from your voluntary swab is indistinguishable as a copy and can become 1 billion copies. We needed to assure people that when they give a swab, it'll be safe. And that's because I do forensic work. We specialize in freeing innocent people. And I get a lot of letters saying, I gave my DNA to the police. Now my DNA's on the knife. And so we made concatenators of these prime DNA sequences that amplify better than human DNA. And we've applied them as a marker to the swabs that you might give to the police. Hasn't been licensed. Disclaimer, I haven't made any money from this. You want to talk to me about it? I'd love to. This is just a paper we published that shows, this is a human DNA profile on the graph called an electropherogram. The height of the peak is the amount of DNA that's someone in my lab. And that's our Nullomer barcode that amplifies quite well. This is a spot from a knife diluted 100,000 times on top and then diluted 1 million times where the human DNA no longer copies because there aren't enough copies, but our Nullomer tag does. A second use that we've put forward for Nullomers is death. This came from the idea that evolution is the longest running experiment on Earth. And if there are forbidden lethal words, we would have detected them. They would be absent. And fortunately, we can synthesize those. So what do biologists want to kill? Pathogens and cancer. Both of those can be recognized by hallmarks that are absent from body cells. That's how the immune system works. And we want to know, do these absent peptides have any effect on these things we might want to kill? So we've published 2 papers looking at 2 of our candidates, 9R and 9S1. They work great in the dish. They kill the 60-something tumors we have in lab, with the exception of 5 or 6 of them. They do quite well. And we have a control Nullomer that doesn't do anything. So it's not like any small sequence of these peptides will kill. Just to show a picture, because it's fun, these are controlled cells. LNCAP is prostate cancer, the first 2 of the columns, and then a breast cancer line, the other 2. And what you see is a low magnification picture, then a high, the controls without any treatment, the control Nullomer, which is one of our peptides, 124, and then our 2 candidate drugs blowing to smithereens these cells. It's gotten some attention. ESPN came to our campus. I was very excited because they have a cancer foundation, and they came to the lab and we talked, and they put this up with our blue field, which is what everybody remembers from Boise State. And so I applied, and they're like, no, you're not eligible. We only give to certain institutions. Such is life. Finally, I'd like to talk about an application of absence that focuses on actual lives and people whose lives we've freed from prison. The axiom in forensic science is that the absence of evidence is not the evidence of absence. This was obviously written by a police lab scientist. If you're accused of crime, the only thing you have is a lack of evidence. And so we've taken cases that have been presented in this way. There's no evidence that they weren't there. And by redundancy of testing many items at the crime scene and showing that it's just one person, not the suspect, we've been able to free more than 24 people. And that's here in the US, in Asia, Europe, and Africa. Most of those clients are people who wanted to prove their absence. This is Christopher Tapp. We helped free him in 2017. He falsely confessed to a rape and murder because he was promised an immunity deal if he just pointed to his friend, who was their real suspect. He pointed to the friend, they processed the DNA from the poor dead woman, it's semen, and it's neither of these guys. They come back to Chris and say, you lied to us, we were sending the deal, and we're charging you with rape and murder. He was tried and convicted, although the DNA didn't match him. We got the case 2007 and pushed for testing of everything we could find at the crime scene, everything he mentioned that they fed him in his false confession. The pubic hairs found on her body, the clothing, and the teddy bear that he supposedly touched, et cetera, et cetera. We had to change the law in Idaho because the Idaho law said you only had 1 year to test DNA. We changed it to any time. And finally, we were able to free him in 2017. The state made a deal. They still called him a murderer, not a rapist, but I think they were convinced that they had a problem. The question was still, who matches that semen? So I promised the victim's mother we would keep looking, and we had tried some unusual things in this case. Before the Golden State Killer, we were trying to identify the last name of the semen donor, the person who left the semen on the dead girl's body, treating it like an orphan child who wanted to find its birth father. And so through public records, we worked with the Idaho Falls Police Department, trained them how to do this. They got a court order for Ancestry.com, which caused the genealogist to be very upset with us. And we were able to identify a last name of the birth father of the man who left the semen. This is Chris just before he was freed with Jen Cummings, one of our lawyers at the Idaho Innocence Project. That's Chris when he was freed in 2017. And following on the leads that we developed, C.C. Moore in the Idaho Falls Police Department, a forensic genealogist who's going to have a television show about this on ABC, I believe, was able to find a man who was in that genealogy that we identified back in 2014, I think it was. And, but his mother had changed his last name, so he had a different last name than the birth father. Another area that we work on is complex mixtures. So when you have more than one person contributing DNA, this is a case in Georgia, the man is still in prison, but we have submitted to the court new evidence. How do you deal with complex mixtures where there's so many people who contributed to a sample? This is a group sexual assault semen sample. At each of these locations, like D3 in the top left, any of us could give 2 possible peaks. There's more than 2, so there's several contributors to the semen. The woman was attacked by 3 men. So this was evidence that we used at Kerry Robinson's trial, that last slide. And I try to explain the complexity of this, where more than one person contributes DNA. I was explaining to a prosecutor and said, well, it's like if we took a Scrabble bowl and put the letters from your name and my name in, we could pull out 10,000 people. And so saying someone can't be excluded doesn't mean they contributed. The vast majority of people who cannot be excluded did not contribute. And the way we did this was to take the letters from our names and look for other names. This was done by our former poet laureate and I in Idaho, Diane Raptosh. And she came up with most of the names. It's pretty amazing. Okay, so we published a paper by taking Ideal Drawer, an expert in cognitive bias, and I took the data that convicted Kerry Robinson, brought it to another lab, 17 analysts, all trained the same way, didn't let them talk to each other because humans are terrible if you let them have consensus. They will give up their thoughts. And we said, no, you have to come to your own conclusion. We got all 3 conclusions. He's included, he's excluded, I can't tell. And it was 17 analysts, only one of them agreed with the state analyst who put this man in prison. We got a lot of attention, the Economist picked it up, and then the National Institute of Standard Technology did a test in 2013. They gave crime labs, 109 crime labs all over North America, the same DNA data from a complex mixture. They gave them 5 questions, 5 scenarios. The 5th scenario is a bank robbery where there's a ski mask, 4-person mixture made by the lab. And the purpose, this is their slide from NIST, is that how many labs would look at this and say, oh, this is too complicated, we can't do it, we're going to say it's inconclusive. What they found out, this is their slide, is 7 labs excluded the suspect, 76 included and gave a stat. The suspect was not part of the mixture. Now, this was a test, and they did this great video conference before. They talked to all the bosses, they did everything. Well, the authors of this paper, I would see at conferences and I'd be like, when are you going to publish? When are you going to publish? When are you going to publish? They sat on this from 2013 till I raised my hand at a conference like this and called them out. And then they finally published it in 2018 with all kinds of terrible disclaimers. So I wrote a piece for the New York Times calling them out on that. Okay. What was the solution? The solution was probabilistic genotyping, MCMC. Mathematics that some of you understand and do every day, and I just have to rely on. And I'd heard there was this new program that could look at old DNA data called TrueAllele by Cybergenetics. And I approached the guy and I said, look, I've got a case. These guys have been in prison for more than 20 years, from 25 years. I think they're innocent. There are multiple contributors to this sexual assault. There's semen, and I think they're excluded, but they're in prison. And the lab said it was inconclusive. And so we were able to use that probabilistic genotyping on old data and exclude those people. The prosecutor agreed, and these men were freed. So we wrote a grant to the Department of Justice, got that funded, and have freed a number of people using this probabilistic genotyping software. This is the latest that we're working on, and we've overturned the conviction, but he's awaiting, he's still in prison. This is Johnny Lee Gates. He has some competency issues. That's why they didn't execute him. He was questionably competent to be killed, and he was given the death penalty, but we got the case. He's been in for 40 years. We're still waiting to see if the state will appeal or he'll be released. But it was this MCMC program that made it an exclusion. So what is it then about these absent bits that can recombine that is so interesting? This is a piece of art that's a commentary on the fake news. This was taken from an airplane. This is a live shot of the diplomat who testified. This was an accident, but I thought it was art. So this is technology, accident, recombination. When is it art? When I say so. That's the secret. These combinations mean nothing until they're recognized, and that's our job. If you think about it, there's only two places where information, symbolic information, can recombine and be recognized as novelty. That's why I call this the aha. It's the nucleus of a cell where DNA recombines, and the idea of flight has evolved several times, in insects by a set of genes, in birds by another set of genes, in bats by a completely different set of genes. And in your mind is the other place, the only other place where symbolic information can recombine. We've gone out, how many light years looking for other information recombiners. We haven't found any. We don't even find life yet, let alone thinking life. So what does all this mean? Omniscience is here, Santa Claus, God. I was young enough to remember when you had to have faith to believe in those, that there was someone who would track your every move, good and bad, and keep a record. As we just heard, it's happened. So in the age of omniscience, what's left? What is prime? What is important? It is finding what's absent. And I suggest we start with the smallest elements. Chance is the mother of all information, all surprise, all creativity. In large doses, chance leads to a lack of complexity, to equilibrium, death, entropy. So all creators have to control the amount of chance in a system. That's what we're doing with artificial intelligence. In jazz, there's more improvisation than in classical music. In viruses, there are enzymes that improvise more. 1 base in 1,000 in HIV is changed in each replication. In us, it's 1 in 10 to the 6, 10 to the 7. Our cells need to be coordinated, except for the immune system and the gametes we produce. And so we control that novelty in all systems except those two. And so what's the new hunt? I would suggest that the new hunt is for chance. Something pure, a source of chance that is beyond our algorithms. It is truly unpredictable, non-deterministic, and it led to absolutely everything that you enjoy. The best tools will be racing for the best chance, a non-biased system. So in light of that, I think it's time to update the list of simple machines and add the dice. This is the original AI. It was used for centuries before anyone really understood how the mechanism worked. Sound familiar? It was the original black box. But it mimicked chance, and it's as important as building pyramids. Living systems have a very simple die. Nature has decided that 4 faces are enough, A, G, C, T. So as we begin to engineer life and to reduce by extraordinary culling the diversity of species, we'll reap the consequences of far fewer replicators and recombinators. This reduction makes us more vulnerable because every microbe and every pathogen that needs a warm-blooded mammal will have fewer choices. They will have to live on a restricted diet of humans. I suggest it would be far wiser to remember that our family tree has a deep root. It extends to the molten core of circulating iron-red liquid that gives us a pulsing ionosphere of protection and replenishes the continents by continual recycling. All things are not food on Earth. All things are not beauty on Earth. They're not riches for us to harvest. But all are dice, dividing and evolving, tightly packed gears that by necessity have fit themselves together. We remove those cogs at our peril. I don't mean to denigrate our species. I think we are the apex of something very important, consciousness. And I believe that we have a unique and vital opportunity. We live on a middle-aged planet. There's only 2 ways for life to get off. An asteroid strike that would take part of our ice cap filled with ciliates out into space, or consciousness, the only species that willingly left the planet. Mother Nature's investment strategy, where she doesn't favor anything, has done really well. She has doubled her investment uncountable times from that single cell in a warm pool. But this blind trust has limits. It doesn't get there fast enough. It needs investment and risk, which is the thing that we do. We decide. And so I'll leave you with the concept that DNA is the fair dice, symbolic only, that I believe RNA changed to DNA for those dice because RNA has a dog in the race. It can be structural and enzymatic. DNA does nothing. It is useless except for recombining, cannot be corrupted. And so, although there is no direction to nature, and there is no reason for the things we see that are, there may be a reason for the things that are not, the Nullomers. Thank you.